Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the A-Level Biology revision topics, and this is about gene expression and genetics. The first part of Topic 2C is Topic 2C1, which is about gene mutation. The different types of mutations we're going to talk about include point mutations, which include these three here. We'll talk about mutations involving genes as well as mutations involving whole chromosomes. A mutation is a random change that permanently alters the DNA. Therefore, proteins that are going to be made from that DNA are going to be completely different. The point mutations, these occur where one or small number of nucleotides are miscopied. For example, it could be maybe this nucleotide and that are miscopied, or they could be duplicated. So these include, like I've already told you, insertion mutations that we see here, deletion mutations, as well as substitution mutations. Insertion mutation could occur because a base has been inserted at one point. For example, between T and G, base A has been inserted, and that is going to shift the whole frame forward, meaning all the amino acids that are going to be added are going to be changed. For example, this is going to be TAA as in the previous one, and the next one is going to be from TAG, and therefore the whole frame shifts forward, leading to different changes in the amino acids added from the point where the insertion has occurred. For substitution mutations, one base has been substituted for another. For example, base A here has been substituted for base G. In this case, there is no frame shift, meaning there is no forward shift or backward shift. However, that specific codon is going to be affected because the amino acid could be different. But in some situations, there could be no effect if at all the base is in the third place of the codon, because sometimes it does not affect the amino acid that should be added. For deletion mutations, a base is deleted and that is going to shorten the whole frame, meaning it's going to shift backwards, and therefore all the amino acids added from that point could be different, leading to problems. The other different types of mutations include chromosomal mutations, where there are changes to positions of whole genes in a specific chromosome. This is for the whole gene, as well as whole chromosome mutations, where the entire chromosome could be lost or could be duplicated during the process of meiosis. So examples of these could be in Down syndrome, where there is duplication of chromosome 21. Exam questions could ask you to search specific base sequences to find if there are some errors. Also, these same questions could ask you to describe the effects of mutation on protein synthesis. In this case, you should describe in detail the effects, how it affects transcription, how it affects translation, including the codon-anticodon ribosomes, as well as the role of RNA polymerase in transcription. Moving on. The next part is about how gene mutations affect the phenotype. And again, remember, the genotype determines which proteins are going to be produced, and those proteins are going to cause the observable effects in the phenotype. Some mutations may have no observable effects, more so if they occur in the non-coding part of DNA, or if a change in a specific base does not lead to a change in the amino acid that is going to be produced, and that means the resulting protein is not going to be affected. You also need to remember sometimes if it's in the third base of the codon, because the genetic code is degenerate, there could be no effect on the amino acid that is produced. When some mutations occur, they give a genetic advantage. So if there is selection pressure due to environmental changes, the organism may have adaptation to survive and be able to reproduce in order to pass on that specific genetic advantage to its offsprings. However, some mutations are catastrophic, meaning they could lead to diseases, and these diseases could include cystic fibrosis, where the membrane proteins are not functioning properly. There are conditions like thalassemia, when blood proteins don't function properly, and there is another condition as sickle cell anemia. This is a genetic disease which affects the protein chain of hemoglobin in the red blood cells, so they have problems with insufficient oxygen, as well as the sickle cells being trapped in blood vessels, which could deprive some areas of their body tissues from oxygen, leading to organ failure. In Topic 2C2, here we're looking at patterns of inheritance. This is about how genes are passed on from one organism to the next. We need to go through some terminologies. For example, a gene is a sequence of bases that code for a specific protein. And alleles are different versions of a gene. The locus is the location of a gene on a specific chromosome. The phenotype is the physical trait or outward appearance. It includes the biochemical characteristics expressed as a result of interaction between the genotype with the environment. This means the environment can affect the genotype. The genotype is the genetic makeup of the organism with respect to the particular feature, for example, being tall, They are genes responsible for height. Now, the word homo means similar, so homologous pairs, these are matching pairs of chromosomes in an individual. They could carry the same gene, but the alleles could be different. And again, remember, alleles are the different versions of a gene. If something is homozygous, it means it has identical alleles for a particular trait, meaning the alleles are the same. And if it's heterozygous, it means it has different alleles for a particular trait. Dominant alleles are those that are always expressed, or those whose characteristics are always expressed in the phenotype. This occurs in homozygotes, meaning homozygous dominant as well as heterozygous. Then a recessive one is a characteristic which is only expressed when both alleles coding for rate are present. For example, in the sense of small t, both are recessive when they are present and there is no dominant. So the expressed characteristic is going to be due to that recessive allele. Here with visualization, we have the homozygous dominant. Homozygous dominant is when both alleles are the same. For example, on this chromosome, we have the gene for height, which is tall, and on this chromosome, we have the same gene, and that is representing tall, so homozygous for that trait. In this situation here, we have heterozygotes. And again, this is the same location, or these two alleles are found on the same location, but on different chromosomes. This is for being tall, and that is for being short. Because they are different, they are heterozygous. And then the homozygous recessive is when both alleles are recessive on the two chromosomes. So we can see small t. This condition is recessive. The two pairs of chromosomes that are side by side are called a homologous pair of chromosomes. Next, we go to carrying out monohybrid crosses. Let's begin by introducing what true breeding is. True breeding is when the two individuals that are crossed are both homozygous for the same characteristic. For example, here we have pea plants. Some have round seeds and others have wrinkled seeds. So the gametes that we're going to have from here are going to be that and that, while the gametes we have from there are going to be both dominant, that and that. And the possible genotype for the offspring is going to be capital R, small r as we see here, capital R, small r as we see there, and that, meaning they are all going to be heterozygous. Their genotype is going to be capital R, small r, and they are all going to be round-seeded plants because the capital R is present and therefore it's going to be dominant. So though they are heterozygous, presence of the capital R, which is the dominant allele, makes them to be round-seeded. So if we cross the F1 generation, meaning the products from here, if they are crossed in the next crossing, both are heterozygous, so we have capital R, small r, and when these are crossed, we have capital R, which is homozygous dominant, and then we have capital R, small r, these two are heterozygous, and then small r, which is going to be recessive and representing wrinkled seeds. So the ratio of round seeds to wrinkled seeds is going to be 3:1, meaning phenotypically, or the outside appearance is going to have 3 round-seeded plants and 1 wrinkled-seeded, but the genotype is going to have 1 capital R, 2 capital R, small r, and 1 small r. And to explain again, a monohybrid cross looks at one specific trait during a genetic cross. Now, talking about test crosses, these are carried out to find the genetic identity of the species that showed a dominant physical trait. For example, if pea plants are tall, you want to find out, are they tall because they're capital T, or they're capital T, small t? We want to find out if they're heterozygous or homozygous. So we carry out genetic crosses in order to identify the genotype of the parents. To do that, the individuals must be crossed with a homozygous recessive one, because there is a possibility that they are all capital T, and then it means we cannot be able to identify that. But if we use homozygous recessive, and in the first generation they are all tall, then that could be a conclusion that the parents were all homozygous dominant. So for example, here we have the yellow seeds as well as the green seeds, this representing yellow and that representing green. The gametes are going to be capital Y, and small y. Here we see the gametes for green and the gametes for yellow. Crossing, we have heterozygous, and this is going to tell us that the genotype of the parent was capital Y, because we have only obtained yellow seeds. However, if a heterozygous yellow plant is crossed, for example, this one here and this one, the gametes are going to be that and that. This will correspond to that, and that is going to correspond to that. Offsprings are going to be yellow, and then here we have green and green, and that tells us the ratio is 1:1, one yellow and then one green, meaning the parents were heterozygous. We can be able to identify the genotype of the parent when we carry out some of these test crosses. However, to do that, we have to use a homozygous recessive individual when we carry out the genetic cross. Now, the next part is about codominance. This is observable in the AB or blood type among humans. This is a situation in which both alleles that are present are going to be expressed. So in the AB or blood type, O is recessive. If A is present, it's dominant, and if B is present, it's dominant. However, if both A and B are present, then codominance results because both are going to be expressed. And again, being blood type A means they are A antigens. Being blood type B, they are B antigens. Being blood type AB, they are A and B antigens on the blood cells. And being blood type O means there are no antigens on the blood cells. So the first cross is between blood type A and blood type O. Here we're going to see the gametes from the parents are going to be that and that. Here we have that and that. The product we see here, blood type A, because A is dominant, O is recessive, so this is going to be blood type A. That is going to be blood type A. That is blood type A. And that is blood type A. So all have the genotype, which is that, and all are going to be blood type A. If a cross is between blood type B and blood type A, in this case, which is AA, the gametes are going to be that and that, here, that and that. The crosses we have blood type AB, blood type A, blood type AB, and blood type A. So the ratio is going to be 1:1, and the phenotype is 1:1 AB to A. The other side, when they are crossed, we have blood type AB, blood type OB, blood type AO, and blood type O. So in this situation, we have all the four blood types. In the genotype, we're going to have that, and the phenotype, we're going to have that. When AB is present, it shows codominance. Going to pedigree diagrams. Pedigree diagrams are like Punnett squares that are used to show, they should be show, how traits can be theoretically passed on and the probability that different offsprings are produced. So these diagrams, of course, they show members of a family or any breeding programs indicating their gender as well as the genetic conditions that they have. An example is what we have here. Remember, for any pedigree diagram given, begin by looking at the homozygous recessive because those are going to give you some information. So this is a genetic diagram showing albinism in a specific family. These ones are males and females with albinism, and these ones are males and females without. And again, I wrote already on this part, but you can see this one has albinism. And if we take it to be recessive as small a, individual one doesn't have, so they could be capital A small a or capital A. But how did I come to the conclusion that they are capital A? That is because none of the offsprings, meaning individual 4 and individual 5, have the condition. So it means because their mother is recessive, none of the offsprings have the condition. The father was homozygous dominant. Because individual 5 does not have, but the mother had, it means it's going to be capital A small a, and this one here is also capital A small a. And because individual 3 and individual 4 have a child or an offspring that is homozygous recessive for the condition, it means both of them were heterozygous. And this one here is heterozygous as well. And because individual 8 is heterozygous and they have an offspring individual 14 that is homozygous recessive, it means individual 7 too was heterozygous. We already know individual 5 is heterozygous. Individual 6 is heterozygous as well because individual 11, who is their offspring, is homozygous recessive for the condition. Also, because individual 11 has offsprings 15, 16, and 17 who do not have the genetic condition, that tells us that individual 10 was homozygous dominant because none of the offsprings had the genetic condition. So all their genotype is capital A small a. And for this situation, we do not know the genotype of this, but we could conclude that since the offsprings did not have any genetic condition, there is a possibility that this further here is going to be capital A. Moving on. In topic 2C3, we're looking at sex linkage. We're going to begin by looking at autosomes, and these are all the 22 pairs of chromosomes except the sex chromosomes. The sex chromosomes are, of course, the X and Y chromosomes, so in total we have 23 pairs of chromosomes. The females are homogametic because they have 2 X chromosomes. The males are heterogametic because they have different chromosomes. They have X and Y chromosomes. The Y chromosome is smaller and contains mainly male sex information. The X chromosome is larger and calls for all female characteristics. So it carries genes that code for traits like blood clotting factors, ability to distinguish color, and that means sex-linked diseases or most sex-linked traits are going to be on the X chromosome. So going on to sex linkage, these are genes carried on the X chromosome, and these are said to be sex-linked. If recessive or mutant alleles on the X chromosome are passed from a female to their male offsprings, these are going to be expressed in the phenotype. And again, remember, males have only the X and Y, and if there is a genetic condition from the mother, since the Y chromosome doesn't have any other alleles that are similar in order to be dominant over these, it means this is going to be expressed because it's going to be the only allele present, and this genetic condition is going to be expressed in the phenotype of that male. So expect exam questions to give you case studies like sex linkage in Drosophila for eye color, case studies for sex linkage in humans, the red-green color blindness, hemophilia, and so on. And again, remember, in humans, transcription factors and epigenetics work together to control gene expression of multiple genes, and this can affect the phenotype. So looking at some of the examples, here we have the red-green color blindness. In our bodies, the ability to see color is coded for by multiple genes, and a mutation in any of these genes is going to affect the ability to see color, causing the different types of color blindness that are observed among people. The affected people can see the red-green color, but they have difficulty seeing the difference between some of the tones of color. And again, like we said already, this is carried on the X chromosome. It's common in men more than women because, of course, women have 2 X chromosomes, and the other X chromosome could have a dominant allele that can be seen in the phenotype, while the males lack another allele on the Y chromosome. So once the recessive is present on their X chromosome, there is nothing to dominate that. Looking at some examples of crosses as well as a pedigree diagram to show color blindness through a family tree, we have a female who is not affected and the male who is affected. And then this genetic condition is going to be passed along the whole family, as you can see in that pedigree diagram. If this is the genotype of the parent and that, we're going to carry out a test cross. So the gametes present are that and that from the female, which is the mother, and from the male, they're that and that. Here we see this individual and that individual are carriers, while this individual and that individual are going to be normal because they do not have the recessive allele. Then we carry out another genetic cross. Here we have a normal male and a carrier female. So these are the genotypes, and that is the genotype of the male. The offsprings produced are going to be a normal female, a carrier female. We have a normal male and a sick male. Then in another test cross C, we have the parental genotype, which is that and that. The possible gametes are this and that and that. The offsprings are going to have that, and that. So here we see we have a female carrier, we have a normal male, we have a female with color blindness, and then we have a male with color blindness. Hemophilia is another genetic condition in which the proteins required for blood clotting, some of them did not form well or are missing. Most female fetuses with this genetic condition may not survive to be born. And if it's untreated in male, it could lead to death from excessive bleeding if there is injury, or from internal bleeding just due to exercise. So let's look at the genetic cross that could occur in cases of hemophilia. So here we see a carrier female and we see a normal male. Carrying out a genetic cross, the gametes for the male are that and that, while the gametes for the female are that and that. So here we see a normal female, a carrier female, a normal male, and a hemophilic male. So this condition has been passed down from the mother to the offspring male. Topic 2C4, this is about cystic fibrosis, which is a genetic condition. Cystic fibrosis is a genetic condition in which there is a faulty CFTR protein. This protein is supposed to carry chloride ions across the membrane in order to cause the dilution of the mucus. So a mutation in a gene, which is a CFTR gene, leads to formation of a non-functional protein, and this is going to lead to cystic fibrosis. Like I already said, the CFTR protein is a membrane protein that transports chloride ions to the outside of the cells. That causes the movement of water from the inside of the cell to the outside so that mucus can be diluted. If this mucus is diluted, then the cilia can move it easily. However, if the CFTR gene is faulty, the chloride ions cannot be pumped from the inside of the cell to the outside, so they're going to build up inside the cell. This is going to lead to water moving from the outside to the inside of the cell, making the mucus to be dry, and therefore it can be thick and sticky and harder for the cilia to move. Individuals affected by cystic fibrosis have slower gas exchange rates due to the thick sticky mucus between the airways, and it's difficult for them to have good diffusion gradients for gaseous exchange. So here we look at a normal membrane. Because sodium ions move outside the cell and the chlorides are actively pumped inside, the chloride ions have to find a way to get out to the other side to increase the concentration on the outside so that water can leave the cell to the outside to cause dilution of the mucus. This occurs in a normal cell, and when that occurs, the water dilutes the mucus, and then the mucus can easily be moved away by the cilia. However, if there is infection, the protein does not function properly, and therefore it does not move the chloride outside. That means the chloride ions are going to accumulate in the inside of the cell, and water is actually going to move into the cell in order to cause a dilution. It moves in by osmosis to cause a dilution, leaving the outside with less water, and the mucus is going to become dry, thick, and sticky, and the cilia cannot move it, so it's going to remain stuck in that area, and it's going to trap pathogens and other microorganisms that could cause further infection within the areas where this is occurring. Next, let's look at the inheritance of cystic fibrosis. If we have two carriers, and these two carriers have children, there is a possibility of some of the children being infected. So the carriers are going to be heterozygous, we can see for both. Their phenotype shows that they're healthy, but they are heterozygous for the condition, meaning they have a recessive allele. So the gametes are going to be that and that. These are the possible gametes from one parent, and these are the possible gametes from another. The possible genotypes of the offspring is going to be that. We have that, that, and that. This is homozygous recessive for the condition, and therefore that child is going to have cystic fibrosis. So the resultant genotype of the offspring is going to be a ratio of 1 to 2. Two of these are going to be heterozygous, and one is going to be homozygous recessive. So in the end, we have 3 healthy children and 1 child with cystic fibrosis. So this is a pedigree diagram showing the inheritance of cystic fibrosis in a specific family. If one parent is a carrier and we have one that is normal, as you can see from the key here, there is a possibility of having a normal offspring as well as carrier offsprings. And if one carrier offspring has another carrier for a partner, there is a possibility of having a child that has cystic fibrosis, one that is a heterozygous, meaning a carrier, another a carrier, and one that is normal. Next, let's look at how cystic fibrosis affects the respiratory system. And again, it affects the respiratory system, it affects the digestive system as well as the reproductive system. So the first example is dealing with the respiratory system. Cystic fibrosis causes thick, sticky mucus to build up in airways, and when this occurs, the surface area for gaseous exchange in the alveoli, as well as the pathway of air in the bronchi and the bronchioles, is going to be blocked or is going to be decreased. That means less oxygen is going to be exchanged or less gaseous exchange occurs. Because of this reduced airflow into the lungs, the concentration gradient is going to be lowered, and that means the gaseous exchange is going to be at a lower rate. People who are affected with this condition will have severe cough in attempts to eliminate the mucus that builds up. Without treatment, the lungs will gradually fill up with the mucus, making it less effective for gaseous exchange. Mucus becomes bacteria and pathogen laden, and that means it's going to cause other infections within the gaseous exchange system or the respiratory system. You need to remember that mucus has antimicrobial properties, but because the mucus becomes thick and sticky, the antimicrobial properties are going to be lost. The pathogens that are held within the mucus are not going to be destroyed, and the mucus is going to provide a good environment for those pathogens to increase in number in order to cause infections within the gaseous exchange system. So here we look at an example where mucus builds up. You can see this is clogging inside the gaseous exchange system. There is bacterial infection that is occurring here because the mucus contains the bacteria. The mucus has lost its antimicrobial effect, and that means the bacteria are not destroyed. They're going to increase in number. They're going to be feeding on the mucus, and they will cause infections within the lungs. And this is all going to affect how the lungs function, leading to less gaseous exchange. Now, going on to cystic fibrosis and how it affects the digestive system, we know that the digestive system produces digestive enzymes, and these enzymes are lytic enzymes, meaning they'll break down specific substances. So to prevent digesting the tissues of the digestive system, mucus is produced in most of these areas so that the mucus can act as a surface onto which these enzymes are released. Now, if I take you back to the previous page, let's see here. Some digestive enzymes are produced in the pancreas. We know the pancreatic enzymes. When they are produced, they go through the pancreatic duct into the duodenum. There is mucus secreted within the pancreatic duct, and if this mucus becomes thick and sticky, it means it's going to clog the pancreatic duct that the enzymes cannot go through towards the duodenum. The enzymes that are released are going to begin to digest the tissues around the pancreas, and if this leads to the digestion of the cells that produce insulin, it means this condition is going to lead to diabetes. There is a possibility of diabetes developing if insulin-producing cells are destroyed in the process. Also, because these enzymes cannot come into the duodenum, there is going to be less digestion, meaning the food eaten, the one coming from the stomach as it reaches the duodenum, it is not going to be digested into the products that can be absorbed, and that means less absorption is going to occur in the small intestines, and this person is going to receive less nutrients. Another thing is there is also mucus that lines the walls of the gut, and this is to protect the gut lining, like I already said. However, if this mucus is thick and sticky, it's going to clog the villi. And remember, the villi are the surfaces that participate in absorption of the digested food. So if they are clogged, the surface area for absorption is going to be decreased. There is going to be less products of digestion that are absorbed, and this is going to lead to lower body mass for the patient suffering from this condition. Moving to the effects of cystic fibrosis to the reproductive system. Again, the reproductive system produces mucus. This mucus changes as the menstrual cycle changes. So during ovulation, the mucus is going to be thinner. The thinness of the mucus is to allow the sperm to pass through the cervix towards the oviduct for fertilization to occur. If this mucus is thick and sticky, it can actually block the cervix, and also it makes it hard for the sperm to swim from the cervix towards the oviduct, and that means fertilization is going to be affected. In males, there can be blockage of the vas deferens. This is the tube through which the sperm can pass. So if this tube is partially blocked, then fewer sperms are going to pass through it. But if the vas deferens is completely blocked, there is a zero chance that the sperm is going to leave the male towards the female, and therefore fertilization will be prevented. Now, cystic fibrosis also has some effects on the sweat glands. It leads to production of salty sweat. This also involves the CFTR gene. However, this CFTR gene works different in the sweat glands than it works in other normal body cells. In other normal body cells, its job is to pass chloride ions out, but in the sweat glands, it's to pass chloride ions in so that there is more reabsorption of chloride ions, taking them away from the sweat. But if it's affected, it means the chloride ions are not going to be reabsorbed from the sweat, and therefore a lot of them will remain in the sweat, making the sodiums also remain there, and the sweat is going to contain excess of the sodium as well as the chloride ions. Normally, the sweat passes along the ducts of the sweat gland. Reabsorption of the salt occurs due to the CFTR protein. Like I've already said, the chloride ions are taken up into the cells, and the sodium ions are going to follow along a concentration gradient. So if the CFTR protein is not functioning properly, there is going to be more chloride and sodium ions remaining in the sweat, and that will mean that the sweat is going to be salty. Also, there is going to be a loss of sodium and chloride ions from the body, and this is going to affect other body processes that depend on sodium as well as chloride ion concentration. For example, nerve impulse transmission, as well as the way the heart functions. When we move to topic 2C5, this is about genetic screening. Genetic screening is of three types. There is prenatal, this is during pregnancy. There is neonatal, at birth, and there is pre-implantation, this is before pregnancy. Each of these is carried out to find about genetic defects that could occur to the fetus or to the child. The first one we're going to look at prenatal is carried out during pregnancy, and it includes amniocentesis as well as chorionic villi sampling. In amniocentesis, they sample the amniotic fluid, and here they sample the chorionic villi tissue in order to determine if the fetus has some genetic defects. The first one I looked at is amniocentesis. Here we can see there is an ultrasound probe that is guiding the doctor as they insert the injection through the womb in order to get sample of the amniotic fluid. This amniotic fluid contains cells that belong to the fetus. Some of the cells are going to be from skin cells. So after this sample is obtained, centrifugation is going to occur in order for the cells to separate from the clear fluid. Both the fluid and the cells can be used. The cells from here can be cultured on a petri dish. After 2 to 3 weeks later, a karyotyping DNA analysis can be carried out. Also, from the amniotic fluid sample, they can carry out other biochemical tests in order to make conclusions on whether the fetus has some genetic defects or not. For chorionic villus sampling, we can see a catheter as well as a vaginal speculum. It is inserted inside and then they take part of the chorionic villi. Because we obtain a lot of cells from this method, it's easy to analyze and you can get the results the next day after the test, but it has some complications relating to miscarriage. Moving on to pre-implantation genetic diagnosis, this is carried out before pregnancy, and in this case, in vitro fertilization is carried out. Hormonal treatments are given to the female so that many eggs mature in the follicles. If many eggs are matured, it means they can be able to extract a lot of mature oocytes that can be used for fertilization. So the oocytes or secondary oocytes are removed using a syringe, and then fertilization is carried out in a test tube because also samples are taken away from the male. So fertilization occurs here, and then the zygote is allowed to divide in order to produce embryos. Samples from the embryos can be tested for genetic abnormalities. Those that do not have the abnormalities can be implanted into the female, and those that can be implanted later can be stored frozen for later use. The storage occurs in liquid nitrogen, as we can see here. So the sample is placed in, and then we have the liquid nitrogen to provide a cooling environment. Now, all these methods have certain difficulties that are associated with them. Number one, we have ethical issues. Of course, this could be due to religious beliefs or cultural beliefs or societal beliefs. Some parents accept the fate of their children, no matter whether the children have genetic difficulties or not. They are ready to take care of them. There are some ethical reasons related to false positives as well as false negatives. False positives are when the test shows that the person has a genetic defect, actually when there is no genetic defect. And false negatives are when the person has a genetic defect, but the test shows that the person does not have a genetic defect. So all these have problems. Here, there is worry that can occur with the parents. The parents can prepare, and after birth, they realize that the child was normal. There is also a possibility that some people could abort the child when the child had no genetic defect. So all these are problems associated with some of these tests. For amniocentesis as well as chorionic villus sampling, there are higher risks of miscarriage. So a counselor should talk to the parents before they carry out some of these tests, because there's a chance that the fetus could be miscarried even when there are no genetic defects. When we look at the bright side, screening allows the parents to get in terms with the fate of their child. It allows some parents to mourn the child that they thought they were going to have. Usually most people think their child is going to be perfect, but if the test is carried out and there is a genetic defect, it allows them to mourn, go through a process of healing so that they can then come to terms and prepare to take care of the child that has a genetic defect if this child is born. So it allows them to reach a decision and make sure that they can be able to either terminate or continue with the pregnancy, keeping in mind that they can be able to take care of the child without any problems. So for most of the situations, parents have to go through genetic counseling, sometimes to deal with traumatic situations related to results from these genetic tests, because it's hard for some parents to take a loss of a child. So this brings us to the end of topic 2C. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.